Alright guys, so this lesson is going to be about meiosis or meiosis. And so far we've learned about the cell cycle and how we make new body cells. And now we're going to learn about how we make a different type of cell. So first of all, I'm going to say that I do not say this correctly. I say it meiosis. It's technically meiosis, and the reason I choose to say it differently is because meiosis sounds way too similar to mitosis. So you can say it however you want, but if you watch videos and stuff, it's probably going to say meiosis, whereas typically I say meiosis. Now this is going to be pretty essential right here. It's a reductive division, and so we're going to be focusing on that concept. And so I do want to do a quick comparison. We have some important vocabulary in this set of notes, and really kind of one of the essential things to understanding meiosis is to understand the vocabulary. And so first off, we need to talk about what somatic cells are. And the prefix soma, soma means body. Okay, so literally somatic cells are your body cells. And almost all of the cells that make up your organs and all of your parts, in fact, all of the cells that make up your organs and parts are going to be somatic cells. They are body cells. And body cells are diploid. Now, diploid, ploidy refers to chromosome sets. And di means two. We represent diploid with two N. So what this literally means is two sets of chromosomes. And remember that you are half of your mom and half of your dad. So you got one set from your mother and one set from your father. And so for us as humans, our diploid chromosome number is 46 or 23 pairs. So we have two of each chromosome. They are numbered 1 through 23. We have two number 1s, two number 2s, two number 3s, two number 15s, two number 20s, and so on. And so the way we get our normal chromosome number of 46 is by getting 23 from mom and 23 from dad. Now, our somatic cells, or our body cells, have this full chromosome count. All of the cells in your body should have 46 chromosomes, and it's your somatic cells that, if they are able to go through the cell cycle, remember that some don't, like muscle and nerve, but if your body cells are constantly going through the cell cycle, they're going to divide by mitosis. They're going to go through PMAT to get two new nuclei and cytokinesis. And in the cell cycle, we were getting two identical daughter cells, and they remained diploid. So we started with a diploid body cell, and we ended with two genetically identical diploid body cells. Now the problem is that when it comes to sexual reproduction, if we were to join two somatic cells, you end up with a total of 92 chromosomes, and that is not a human chromosome number. And so we're going to need a different type of division. And so this is where meiosis comes in. So this is a reductive cell division, and it's called reductive because it is reducing the number of chromosomes. And so instead of making somatic cells, meiosis makes gametes or sex cells. So these are pretty much the only cells in your body that do not have that diploid number. For guys, this is going to be sperm. For girls, this is going to be eggs, okay? And so meiosis is a reductive division that's going to reduce that chromosome number by half. And so the cells that are generated are gametes. Instead of being diploid, they are going to be haploid. And so you can always think of haploid as half. So diploid was two sets of chromosomes, one set from mom, one set from dad. Haploid is only one set of chromosomes. So essentially you can think of this as either mom or dad. So where your diploid cells were mom and dad chromosomes together, your haploid cells are either mom chromosomes or they are dad chromosomes. And so this way, when reproduction occurs, you have 23 in the egg, you have 23 in the sperm, and that's going to create that normal chromosome number of 46. Now there's actually two parts to meiosis. Okay, there's meiosis 1 and meiosis 2. You're going to see a lot of overlap between mitosis and meiosis, but I will tell you that this right here, meiosis 1, is where all the differences are going to happen. Okay, meiosis 1 is going to be the stages where some very significant events occur that we did not see in mitosis. Now, if we look at what's happening, meiosis 1 is separating homologous chromosomes. We're about to talk about what those are. Meiosis 2 is separating sister chromatids. Now this is what happened in mitosis. And so meiosis 2 is like mitosis. So out of the two, when we look at meiosis 2, I don't know why the highlighter does that. When we look at meiosis 2, we're going to see that it is very similar in the processes to mitosis. So it is like mitosis. Now, if we look at the outcome of meiosis, we're going to see that it's pretty different from the cell cycle that we've already covered. Whereas with the cell cycle and mitosis, we got two genetically identical cells, and they were both diploid. Here we're getting 4, so we're getting double the number of cells because we're going to go through a series of 2 divisions. They're going to be different because of some significant events that happen up here in meiosis 1, and they are going to be haploid. We are going to divide that chromosome number in half. So the outcome of meiosis is going to be significantly different from the outcome of mitosis and cytokinesis. Now, in your bodies, this is happening in those reproductive organs. So for females, meiosis is occurring in the ovaries. That's where your eggs are produced. And in males, this is occurring in the testes. It is in these body cell or these body organs where you have cells that are called germ cells. And then these are the cells that are going to be undergoing meiosis. All right, so before we get into the processes of meiosis 1 and meiosis 2, we just need to clarify a couple of things, review a couple of terms. So first of all, the word chromosome. Now remember, most of the time throughout a cell's life, the DNA is in the chromatin form, which is the lesser condensed form. And it's only during division that we see the chromosomes, which is the supercondensed form of DNA. The chromosomes are kind of inactive during that time because they're so tightly wound and tightly packed. And that when we see them, the chromosomes typically look like an X because they have already been duplicated. So anytime you see the X, this is super important for meiosis that we understand this. Anytime we see the X, we need to remember that that is a double copy, okay? So it consists of 2 sister chromatids held together at the centromere, and we see the X after the S phase, and we only see them during those division stages. Now, another term to know is homologous chromosomes, and we've seen the prefix homo before, and we're going to continue to see it. Homo equals the same, and so homologous chromosomes are chromosomes that are the same. So earlier I told you that you have 2 of every chromosome. They're numbered 1 through 23, and so you have 2 number 1s. You got 1 from mom and 1 from dad. You got 2 number 2s, you got 1 from mom and 1 from dad. And so that's what your homologous chromosomes are. They are your chromosome pairs, or they are your like chromosomes that you got from each parent. So it's your pairs, your 1s, your 2s, your 5s, your 12s, your 18s, all the way up to 23. Now, they are similar, okay? They are the same size, and they contain the same genes. However, they are not identical to each other, okay? In fact, that would be quite weird if your chromosome pairs were identical to each other, because remember, they came from 2 different parents. So your mom gave you a number 1, which has certain genes on it, and your dad gave you a number 1, which has certain genes on it. But we haven't talked about genetics yet. We're getting close. The thing is those genes can come in different versions. So, for instance, let's say we're talking about eye color. Your dad could have the eye color gene that says brown, but your mom could have the eye color gene that says blue. And so even though they're the same information, they may not be exactly the same. In fact, they are not going to be exactly the same because they're 2 different people. So your homologous chromosomes are the same size and the same information, but that information can vary from your parents. One may say, tall, one may say short. One may say curly hair, one may say straight hair. And so even though they're the same genetic information, there's different versions of those genes called alleles. So while they're the same, they are not identical, okay? And that's important to understand. That's how we kind of get variety through sexual reproduction. All right, so this right here is what we call a karyotype. We actually looked at these probably back in freshman year. But karyotypes are just a picture of chromosomes. Now you can see that these particular chromosomes are the single chromatids. So this was done after they are separated in anaphase. But here you can see there's two number ones, and what you look for on a karyotype, how you put the karyotype together, is they're the same size and they have the same banding pattern. And so you can see there's a pair there, all the way down to pair 23. Okay, so you've got 46 pairs of chromosomes in a normal human, and they are the same size and the same genetic information, same banding pattern. All right, so what we're going to do is we're going to start to take a look at the processes of meiosis, and once again, you're going to see a lot of overlap, okay? So just like we saw in the cell cycle, we are going to have an interphase. Now remember, these are going to be your germ cells that are in your ovaries or your testes. And in interphase, it's going to be the same basic stages that we saw before, okay? It's going to be the resting, growing, doing normal cell stuff. And so just like before, you're going to have your growth phases, your G1 and your G2. And then you're going to see the S phase, just like we saw before. Now, it is important to understand that like mitosis, there's only going to be one interphase that precedes the divisions, okay? So in the cell cycle, it was interphase followed by mitosis and cytokinesis. Now, the difference here is that we're going to have interphase, but it's going to be followed by two divisions, okay? But still, there's only one interphase. We're only going to double the DNA one time, and this interphase is only going to occur one time, and that is before meiosis 1. So following interphase, we are going to enter meiosis 1. And like before, we have PMAT, okay? Now, the difference here is now they are followed by a number, okay? So in mitosis, we just had PMAT. Prophase, metaphase, anaphase, telophase. If those words are followed by a number, then we know that they are meiosis and not mitosis, okay? So now we have prophase 1, all right? It's not just prophase. Prophase is only in mitosis. In meiosis, it's either going to be prophase 1 or prophase 2, okay? So in meiosis 1, we're going to follow all of our PMATs with the number 1. So in prophase 1, we're going to see the basic events that we saw before. Okay, the nucleus is going to break down. That was one of our normal prophase events. The spindle is forming. That's one of our normal prophase events. Chromatin condenses to form chromosomes. That's a normal prophase event. Remember, we've already had an S phase, so those chromosomes are going to be X's, okay? But this right here, you need to definitely understand this, y'all. This right here is key. One of the most important stages in meiosis that you need to know is prophase 1, because this is where something very significant happens that only happens in prophase 1 of meiosis 1. And so if you think about your brothers and sisters, you are different, okay? You may have come from the same parents, same basic genetics, similarities, but you are not identical. And this right here is one reason, with the exception of identical twins, of course. This right here is one reason, one of the main reasons why you are different from your siblings, okay, is because of this process that occurs right here in prophase 1. So this does not happen in mitosis. This does not happen in meiosis 2. It only happens right here in prophase 1. And we have lots of vocabulary just in these two bullets here, okay? So first thing that happens that is different is the chromosomes are going to pair up, okay? Now, this process of pairing is called synapsis. And so we've talked about how we have 23 pairs in meiosis 1, prophase 1. Those pairs find each other. So the 1s get together, and the 2s get together, and the 3s get together, and the 12s get together, okay? And so synapsis is the process of the homologous chromosomes pairing up, okay? So there's one vocabulary word right there. Synapsis is the pairing of your homologs, or your homologous chromosomes. Now, while they're together, we call them a tetrad. Now, tetra means 4, and if you have a pair of chromosomes that look like this, all right, there's 1, 2, 3, 4 chromatids, okay? So the pair is called a tetrad. So the process of pairing is synapsis, and the pair itself is called a tetrad, okay? So your 1s are going to be a tetrad, your 5s are going to be a tetrad, your 20s are going to be a tetrad. Now, this is another important thing, the most important thing. While they are paired, crossing over may occur, okay? Now, crossing over is where they exchange pieces of their chromatids. So let's say this is mama chromosome, and this is daddy chromosome, okay? So mama chromosome has 2 identical chromatids. Daddy chromosome has 2 identical chromatids. Now here's what happens in crossing over. So let's say that mama chromosome was originally green, okay? So this is mom, this is dad. Now, in crossing over, they can exchange pieces of their chromatids. Now, why it happens, I don't know. Exactly how it happens, I don't know. What's important is that you need to know that it does happen. Now, not every pair may cross over, but as long as one of the 23 crosses over, we're going to have unique gametes. And some pairs cross over more than others, okay? But while they are synapsing, they actually kind of overlap a little bit, and they can exchange pieces. Now, they have to exchange like for like, okay? So a lot of times if I'm in class, I model this with another student. So I stand up in front of the class like a big X, okay? And I get another student to stand up in front of the class like a big X. And so when crossing over occurs, you have to exchange an arm for an arm or a leg for a leg, okay? Like, you can't have this top part over here exchanging for this bottom part over here, because on these chromosomes are genes. And so you have to exchange like for like, otherwise you're losing genetic information and you're creating mutations. So what's going to happen is, in the process of crossing over, they're going to exchange like pieces. So a piece of the mama chromosome is going to break off, and it's going to switch with a piece of the daddy chromosome. And so the yellow is not going to cover the green. And so what's going to happen is there we go. What's going to happen is you end up with, no longer do you have identical chromatids. Okay, so here, originally I had identical chromatids in the mom. Well, now this chromatid is different because it has a piece of that daddy chromosome. And then my 2 daddy chromatids, which were originally identical, are no longer. Okay, you can see now that this chromatid is different from the other one. Now, just to kind of give you the big picture, the end result is that these chromatids are going to end up in different cells. So this is going to end up in one gamete. This one's going to end up in another gamete. That one's going to end up in a gamete. And this one's going to end up in a gamete. So now that chromatid is going into a cell. That chromatid is going into a cell. That one's going into a cell. And that one's going into a cell. None of those cells are getting identical chromatids, okay? So every single one of those final cells are going to have a unique combination of genetic material, okay? So I know this is not as easy to draw and explain without a board in front of me, but the main thing you need to understand is that crossing over is going to generate diversity, okay? By crossing over occurring, and like I said, if just one chromosome pair does this, and more than likely multiple chromosome pairs do this, which means even more variety, when they exchange pieces of their chromatids, we're going to generate diversity. This is why you and your siblings are unique, okay? Even though you came from the same parents, you're not getting the exact same set of genetic information. So that is one big area where variety comes from. All right, so these pictures are a little bit better than mine, okay? So you can see here. All right, now here's another vocabulary word for you. Chiasmata is the location where they physically cross over. So when they're pairing up, remember that's called synapsis. The pair itself is called a tetrad. While they are paired, they kind of overlap a little bit, and the location where that crossing over happens is known as the chiasmata, okay? So you have lots of vocab words right there. So once again, homologous chromosomes pair up. The pair is called a tetrad. While they're paired, they may exchange like pieces of their chromatids, and when that happens, no 2 chromatids are identical anymore. And that's going to lead to what we call recombination and variation. All right, so now we're going to move forward. Okay, so that was prophase 1. That was one very significant one. The next one is metaphase 1, and I want you to kind of be familiar with this one too, because there's going to be another difference here. So in metaphase 1, we still have lining up in the middle. Okay, so that's the same. but this time they're lining up as pairs, as tetrads. So in mitosis, when they lined up like this, single file, okay? In meiosis 1, they're going to line up as pairs. All right, so instead of having 46 chromosomes in a line, I'm going to have 23 pairs of chromosomes in a line. So this is only going to happen in metaphase 1. Once again, all of the key differences occur in meiosis 1. So they line up as tetrads this time instead of single file, okay? Now keep in mind that right here we're going to have another area for variation, okay? Because every time they do this, you have a mom and a dad chromosome, all right? So it doesn't matter which one goes on which side, all right? So this lining up with mom and dad, okay, it's not like all moms go on one side and all dad chromosomes go on the other side. It's going to be unique every time, and this is going to be another area for diversity called independent assortment, and we'll take a look at that here in a minute. All right, so metaphase 1, they are lining up in the middle, but this time they're lining up as pairs. Anaphase 1, they're going to be pulled apart. Now this is going to be different because they are still being pulled apart, but this time it's the pairs that are separated, okay? So we aren't separating chromatids yet, we're separating those homologous chromosomes. So now these are going to one side, and these are going to the other side. And in telophase 1, my new nuclei are going to reform, the spindle's going to break down. I'm going to end up with 2 new nuclei, okay? Cytokinesis is going to happen. I'm going to end up with 2 new cells. And so by the end of meiosis 1, I'm going to have 2 cells, okay? 1 became 2. Already, they're not identical, and this is going to be because of crossing over and independent assortment. And already they are haploid, okay? So I want to show you the picture here. So in this picture here, we started with 4, okay? They paired up, synapsis, they crossed over, okay? They lined up as pairs. Now here you can see the darker blue is on top, the lighter blue is on bottom. That could just have easily been the other way around. That's where independent assortment comes in. So in metaphase 1, they lined up as pairs. You can see that crossing over is there, okay? Then in anaphase, the homologous chromosomes were pulled apart. And in my end result, I have 2 chromosomes in each cell, okay? So I started off with 4, I ended up with 2, okay? So already I'm haploid, okay? So my end result is 2 non-identical haploid cells. Now you might be thinking to yourself, if you're haploid, why are we not done, okay? Now this is where you have to understand what chromosomes are. Look at what these chromosomes look like. They are X's. And anytime you see an X, you should know that that is a double copy. So even though these have half the number of chromosomes, there's technically half. If this were our cells, there would be 23. But the problem is they're duplicated chromosomes. So there are 2 copies of every gene. So even though there's a half the number of chromosomes, there's double the amount of genetic information, because every chromosome is a doubled copy. So our next phase, meiosis 2, is going to be about separating those sister chromatids so that they are truly haploid and don't have double copies, okay? Because remember that this is a doubled chromosome. When we separate them, each sister chromatid is their own chromosome, okay? So I know that word can be kind of confusing, but the reason we have to keep going is because right now we have double copies of every gene. So meiosis 2 is going to be all about separating those sister chromatids. Remember, this one is going to look just like mitosis. Now, we're starting with haploid cells that are different, but in terms of the processes, in terms of your PMAT, this is going to look just like what we saw in mitosis. So in prophase 2, the nuclei break down, the spindle reforms, we see the chromosomes. Metaphase 2, chromosomes line up, this time single file in the middle of the cell. Anaphase 2, we're going to be pulling the sister chromatids apart. So this is what we did in mitosis. So once again, we're pulling them apart. And then in telophase 2, we're getting 2 new nuclei. This is going to be followed by cytokinesis. And this whole process is going to look just like mitosis. So here's my original cells. I'm starting with 2 this time. They line up in the middle, chromatids are pulled apart, and by the end, I now have truly haploid cells. Okay, we no longer have a double copy of every gene. Okay, so what I want you to notice though is if you look at my 4 nuclei, no 2 cells are exactly the same. Okay, so like this one has a big light blue and a small dark blue. This one down here has a big dark blue and a small light blue. These have the crossing over bits on them. Okay, so the more chromosomes you have, the more crossing over you're going to have, the more independent assortment you're going to have, and the more variety you're going to have. And so the end result, each of these is going to be a gamete. Okay, the end result is that all 4 gametes are going to be unique. All 4 gametes are going to produce a different baby. And so this is why it's really kind of interesting to think about. I know you guys are several years away from having kids, most likely. But when you do have children, it's kind of interesting to think that every child is a unique combination. There's only one way that you could have gotten that child, okay? There's only one way that I could have gotten my Gracie or my Paxton or my Addie, okay? Any other gamete, and it would be a different child, okay? So you truly are a unique combination, and you truly are a special individual because there's only one combination that could have produced you, okay? And it all goes back to meiosis and the production of these sex cells. All right, so the end result is we have 4 different haploid daughter cells. Remember, that's very different from mitosis. So they are 4 because we had 2 divisions. They are different because of crossing over an independent assortment. And they are haploid because we divided those homologous pairs, okay? So what causes the differences? That independent assortment. So remember that in metaphase 1, they line up in pairs. It doesn't matter which one goes left, which one goes right, okay? So that's going to be a unique lining up, and that's going to lead to a unique combination of mom and dad chromosomes. And then we also had crossing over that occurred in prophase 1. And between the 2, every gamete is going to be a very unique combination. Now it is a little bit different in males and females, okay? In males, all 4 of the gametes are going to be sperm that could be used to produce a baby, okay? Potentially every time those germ cells undergo meiosis in the testes, you're creating 4 sperm that could be used to make a baby. Now this isn't in your notes, but I also want to tell you a difference in the timing, okay? So for guys, this process starts at puberty. Sperm production occurs once puberty sets in and those hormones kick in. And essentially, guys will produce sperm the rest of their lives, okay? You may have a decline as they get older or there's changes in testosterone. But for the most part, guys become sperm factories at puberty and they remain sperm factories until they pass away, okay? This is why you can see older men having babies, okay? Now, in females, it's a little bit different. So in females, they don't get 4 eggs. For every meiotic division, they get 1 egg because 1 cell gets most of the cytoplasm. During cytokinesis, it doesn't divide equally. And so what happens is you end up with 1 really big cell, 1 egg. And the other ones, while they have the haploid chromosomes, they don't get all of the extra cytoplasm and organelles. And so we call these other 3 cells polar bodies. Now, this is kind of important because from literally day 1, the mom is kind of the nurturer, okay? Because what the sperm is going to provide is the other half of the chromosomes. The egg is going to provide all of the stuff that zygote needs to grow and divide. So all of the energy for mitochondria and things like that. This is also why you inherit your mitochondrial DNA from your mom primarily. It's because that egg is going to have all of the things that it needs to go through divisions and go through development. So the sperm pretty much drops off the chromosomes and it's done. So one of the cells gets the majority of the cytoplasm and organelles. That becomes the egg. These polar bodies, while they have the chromosomes, they can't really be used to make a baby. So guys get 4 sperm per meiotic division. Girls get 1 egg and 3 polar bodies per meiotic division. Now, another difference is the timing. Like I mentioned, guys start at puberty and they continue through life. Girls, you actually start this process before you're ever born. Your eggs are partially formed in the womb, okay? And so a lot of times you hear jokes or people talk about, women, their eggs expiring, or you talk about, they'll look at egg counts when it comes to fertility. And that's because you're born with all of the potential eggs that you're going to have. And as you live, those cells are slowly dying. So that's why you won't see older women necessarily having children. Number one, a lot of times after they go through menopause, they can't. But number two, the older you get, the fewer viable eggs you have. And so your chances of reproduction go down. You also see an increase in the chances of genetic abnormalities in older women. And so it's all because those eggs have been around since before you were born. So pretty much you go through about meiosis 1 before you're ever born. And then once a female hits puberty, then they start to release an egg, and it will go through the remainder meiotic divisions. And so the older the woman, the older the cells, okay? And so a lot of times there's some difficulties there when it comes to having babies as a woman ages. So just to kind of, tell you those differences, you don't necessarily have to know about the timing, but I wanted you to be familiar with that. All right, so in the crossword puzzle you have in your study guide, you don't necessarily have to do it, but there's a couple of clues, and one's about sperm formation. It's called spermatogenesis. The other one is about egg formation. It's called oogenesis. Usually anything related to eggs starts with an O, ovary, ovum, ova, ovary, but oogenesis is the process. Now, technically this right here would actually undergo another division, and I would get 2 more little polar bodies, okay? So you can see that guys get 4 viable sperm, girls get 1 egg, and 3 polar bodies. All right, so a quick side-by-side comparison of mitosis and meiosis. Now, technically, this would be the cell cycle and meiosis as a whole. I want you to notice that they start the same. They both begin with a diploid cell. This would begin with a diploid cell in the body. This would begin with a diploid cell in the ovaries or the testes. Okay, so that's going to be the same. Both of them go through an interphase where DNA is replicated. So there's one S phase, one interphase that precedes each process. Now the end result is going to be very different. Okay, mitosis is going to divide once, PMAT. Meiosis, there's going to be 2 divisions, PMAT1 and PMAT2. Now in mitosis, I'm getting 2 identical diploid cells. I didn't have crossing over. I didn't have independent assortment. I didn't have the chromosomes pairing up in metaphase. And so I'm getting 2 identical diploid, whereas in meiosis I'm getting 4 different haploid because of synapsis and the way they pair up and independent assortment. Now, mitosis and cytokinesis is a form of asexual reproduction because I'm getting the same. Whereas meiosis by itself is not sexual reproduction, but it does create the cells needed for sexual reproduction. Because sexual reproduction is about 2 parents, and so in order to have information coming from both parents, we need meiosis to produce those sex cells. So once again, asexual reproduction, it only involves 1 parent, okay? It does not involve the fusion of gametes. So this includes mitosis, like our cells will go through, our body cells. It includes binary fission, which is what bacteria do, and there's some other asexual processes like budding and grafting and things like that. The new cells are identical. This is the key to asexual, is you get the same. And so you don't get genetic variation, which could be detrimental when it comes to natural selection and evolution. Sexual reproduction is superior because you do get diversity, and diversity is always better. This involves your gametes and the process of fertilization. And once again, we know that every offspring is going to be unique, okay? And so that's why sexual reproduction is better, because the more variation you have, the better off you have survival. Now, I just want you to kind of understand a little bit here. One of the things we're supposed to cover are the different types of life cycles, and we'll go into some of these in more detail in second semester. Like, we're going to talk about alternation of generations. But what I want you to understand is that meiosis and mitosis are not created the same in all organisms. Okay, the basic processes might be similar, but the timing of those processes might be different. And so for most animals like us, this is our basic kind of life cycle. So you have an adult, okay, a diploid adult. They are going to produce sex cells by meiosis. Those sex cells come together in fertilization and they produce a zygote. And then that zygote, which is diploid, is going to go through mitosis to produce the adult, okay? And then that cycle will continue. Now, it's a little bit different in fungi, which I'm not an expert in fungal reproduction, nor do I care to be an expert in fungal reproduction. But it's actually different here because they actually don't have male and female. They have like plus and minus. But their zygote will be diploid, but then that diploid zygote goes through meiosis. And so the adult cells are actually haploid. And then they go through mitosis to produce the gametes. So same basic processes, just different order. Okay, you don't have to memorize this. I just want you to be familiar that not all life cycles are exactly the same. Now with plants, they have something called alternation of generations. Once again, we're going to go into this in great detail when we get to plants. For now, what I want you to understand is that they actually alternate between a diploid stage and a haploid stage. It's called the sporophyte and the gametophyte, okay? Now, the sporophyte, which is diploid, is going to produce haploid spores by meiosis, and then those spores go through mitosis to produce the gametophyte, which is the gamete-producing structure, and then those are going to go through mitosis to produce the gametes. So like in us, we're producing gametes by meiosis. Other types of organisms may produce those gametes by mitosis. It just is a different type of life cycle. Okay, so all you really need to know about this is that plants are going to perform, or they have a life cycle known as alternation of generations, where they alternate between a diploid and haploid stage. And we will go into the details of that when we get to Kingdom Plantae. All right, and that's going to wrap up our lesson on meiosis. Okay, I hope you guys understand that as well as you can. I would encourage you to do your study guide for this unit. I think that can be very helpful. And next class, you're going to be doing an activity to hopefully help you see the differences between mitosis and meiosis. And so if you have any questions, just let me know. And I hope you guys have a great day.